how to prepare, what are the process to improvement of the herbarium. So these herbarium concept and techniques, the plant collections are essential for taxonomy researchers because they serve as a voucher specimens. The specimen vouchers which can preserve in the laboratory of botany or university level and departmental or degree college level, and also called you say the national level, international level. They also help to the identity, the family, genus, and species, which herbarium is a basically storehouse of botanical specimens, storehouse which we are observe our identity base to collection of the present herbarium, which are arranged in the sequence of an accepted classification system, which means Bentham and Hooker basis classification to prepare the herbarium in India and available for references to other scientific studies also there in the herbarium specimens which are stored in the laboratory of botany in the department of botany or degree colleges. Once mounted and deposited in the herbarium, the collections are refers to the herbarium specimens, such as herbarium specimens can be stored for many years, and as such, they are as historical collections. First point. Second one, references collection for checking and identity of the newly collected plants. Checking and old collected plants and newly collected plants both are also called store, that is called herbarium importance as an aid to teaching and as a source of research material. It is also teaching aid, one of the teaching aid of herbarium, how to call the name, by how to identify the plant's name from environmental or surrounding areas of the colleges, and these are materials required for plant collections. The plant cutter, pruner, scissor, knife, leather glues, field notebook, pencil, permanent ink pens or magnifying glass, digital camera, flora book, colorful or process, forceps, which are all materials. Then newspapers, also bladder, herbarium, pressure, sharp and straps, tissues, paper, plastics, bags, herbarium bags, which are all is also material required for collection of the herbarium specimens, also living specimens. Then the herbarium specimens are collected, drying and press the neat herbarium sheet. That drying table, mountain, survival blankets, clipper, heater, that is also one material or few materials. First aid box, also called a topographical maps. Then microscope, also called a computer and GPS system. It is a present type of the material for preparation of the herbarium. Then first processes are collection of the specimens. The twig with a good flowers need to be collected for the specimens. The portion of the specimen should have a contain clear phyllodes, and the branching system, it is a one part for small herb collection of more specimens, and also fixed herbarium sheet up to 6 in the decimal, also desirable plant parts which can collected first, and also this part is twig pressed on the newspapers. In general, scissor, plant cutter are used to cut the twigs, while for a bit height or a down or pruner is used, and for the spiny specimens a leathery glue is required. Then spiny for ferns and herbs are digger is applied to take out the underground portion of the plant. That is one important process for collection of the specimens to prepare the herbarium. The aquatic plants are filmy or somewhat filamentous, also filamentous, and are difficult to be arranged on the sheet. So these plants need to shaken well before putting herbarium bags. These bags. Some plants can be put in plastic bags. That is called a shaken pot. The soak with a blotting paper before putting the sheet of a mounting paper, and then paper is lifted, making a slope, then carefully so that it is a facilitated water runoff and clean the surface of the area of the twig. Then diseased plants, infected twigs, etc. should be avoided. That is also one process of collection. The second one is also field note. After specimens, then collection of the field record is noted. A small pocket-sized notebook which can keep to the pocket of human beings or students. The good quality of the specimens also become worse. It is a does not have the good field record. Then one important, then date of collection, location, name of place, or distance from definite point. That is collection number, if possible, name of the specimen and description for the floral parts that may change after drying and noted drying. After drying, that plant change, then note down. Then a topographic map is essential for the location. The topographic is important. For the characters are should tally with the literature. Then Gambler and Smith and Gambler flora bases to identify the name of the plant, generic name and species name correctly of the book. Dorsal and ventral leaf views or photos are also essential for further identification, confirmation. It is also one. Then it needs to examine the floral parts carefully. It is small using so imagination for the lens. Digital camera can be used to close up the image, even to the flower too small or the flower and leaves, which can take to the digital camera with the digital camera to take the need to photograph, which for the flora are also called a manual book. Then likewise, the specimens of the microhabitat means associated specimens should be mentioned at least 5 species. Finally, with distributed status of plant also need to be the mentioned either the collected species is rare, that is important. Then the frequent, common, locally common or occasional plants which are collected in the first process collection of the specimen. The collection number can be used as specimen tag. These number usually begin with 1, 2, 3, or etc. and continue indefinitely number based on the degree and intermediate students. Degree maximum 50 specimens are required for preparation of the degree level collection of the herbarium. For students, all above information is required during the preparation of a sheet with the specimen. So this is also third process or pressing. The specimen should be placed the field press to the first opportunities, either so directly with the collection or sometimes are say after a temporary storage in the vasculum. That is also first point. Then second one is the specimen is also the shorter than 15 inches, 38 centimeter each specimen. Particularly, it again should be kept and directly in the folded, without folded, or a newspaper after loosely also called spreading the leaves and also branches, herbs, which are a generally collected process. then also along with the roots, and also longer than the 15 inches. The longer the roots are also one important for the preparation of the say herbariums. So then it can be located in the form of the VRNRW type of the book. Unnecessary overlapping leaves and other parts must be evolved. So then large leaf of the pinnately compound leaf sometimes are which are available in a pinnately compound. So a branch or a so only kept. A few leaves may be turned over a show lower and upper views. Also based to the preparation last if the specimen is a gymnosperms where leaves fall before die after falling and before dry. So the specimens are needed to the deep in the glycerin also before pressing. Glycerin is also pressing in only gymnospermic plants. It is also called a repressing. The fourth process are repressing. The specimens are repressed in the also called evening in the camp of the institutes, which institutes that colleges also got a university that is also based to the preparation of the parts of the plants. So the biodas or the bladder are changed in the they are a moistened moist the moisty leaves when dry after dry which are causes of the fungus. The specimens are kept with a new bladders and the old bladders to new bladders are shifted when the leaves are which are completely dry. After it can press to prepare the herbarium mounted on the separate sheet. The herbarium pressure equipment can be made from the wood scraps, the old cardboard boxes, old newspaper, and small ropes. It is also important for the repressing of string. We can prepare the herbarium. So the final data is recorded in the computer using the Microsoft Excel program before specimens represent for a drying in the field. Thus, the prepare the database is a helpful in future. In future, that is also computer based. Also, Microsoft Excel program. It is a one important process. Then also fifth one is a drying. So these drying techniques are of the two types. Those are accomplished without heat and those with the aid of the artificial heat with then the accomplished without heat and artificial heat, the two type of the process which can use to the drying process. The during also natural drying changes, the newspaper is also so necessary natural, but without effect of the twig. We can use only natural process for drying of the twig with can use of the newspaper. It also necessary and the plant should be carefully transferred from one newspaper to another. That is also one important without any damage of the twigs. We can transfer from the old newspaper to new newspaper. So the change of the bladders are newspaper. So sheets are repeated every few days, then increasing the intervals between the changes. So then successfully until the specimens are fully dry. So these fully dry specimens are which can fit or successful part of the twig to prepare the herbarium sheet. It is a mount on the new herbarium sheet. Then the whole process of the drying so may take about 10 days to 1 month. That is time is important. Not only based on the examination for submitting the herbarium record to prepare. That is a non-complete, not successful process. Only the preparation is which can we can use to the permanent preparation of the herbarium. it can use of the up to 10 days of the dry of the twig, then up to 10 days to 1 month. So depending on the specimens and the climate of the area, for example, xerophytic plants. Then also got the hydrophytic plants, which can have more moisture in nature of the plant or twig that can may use up to 1 month of drying period. So they press, the press is kept in the dryer and a cabinet, which is a lamp or a electrical bulb. The lamp also electrical bulb, which is also artificial drying process. So the drying process are we must use the two types. You know that the first one is also called a accomplished drying, also called electrical drying. The electrical drying is which you put on the under the shade of the light. Then also the press kept in the dryer cabinet, which is a lamp or electrical bulb, warms or heater. That is one. The heater drying with the help of the artificial heat takes 12 hours to 2 days, maximum 2 days, and 12 hours are minimum day time period for dry of twigs for preparation of the herbarium process. So then last point is sixth is a mounting herbarium specimens. What is a mounting? Mounting is the process by which specimen is attached to a herbarium sheet and a label to affixed or fixed at the lower rate of the corners. Then the total complete herbarium sheet only lower space. The specimens are mounted on sheets of a standard size herbarium papers, 29 into 43 centimeter each sheet. That is also maximum sheet for degree and also intermediate fraction. The most herbarium use a glue or paste or paste to fasten specimen to the sheets. The paste are also called a glue. It is a most important. The small paper envelope called the fragments, packets are attached to the sheet, to the whole sheets, extra flowers or any part of the specimen is the additional support of the additional matter of the herbarium. So these are believed to the valuable because of providing long-term protection against the insect attacks. Insects are attacks. So there's also called a thymol is a quite effective of the fungicides. Thymol, this the thymol powder which are a spray on the twigs, which can protection of the fungus. So then precaution should be taken to the protect herbarium specimens from the damage by insect or pest or microbes. Insect, pest or microbes protector, that is called a thymol is a quite. Then also called a fungicide. Then next one, insect, also called a repellent, such as naphthalene balls. The naphthalene balls, which are important of the process for or naphthalene ball or para-dichlorobenzene. The para-dichlorobenzene are sometimes are placed in a small quantities of the herbarium cabinet. It is one, but all through dangerous and so hazardous of health. Mercury chloride is also one part. It is so. This is a process of the production area, which then the uses of the plants are herbarium. Then eighth one is also labeling process. So the an introduction also label in essential part of the permanent plant specimens. Essential. So it is a primarily contains the information recorded in the field diary, that primary information and that habitat, that area, which areas are available or obtain that process. So at the time of the collection, the best position for the main label is generally through to be a bottom right side. bottom right side. The following information is required for labeling during the preparation of the sheet with the specimen of herbarium. The Department of Environmental Sciences also called it, then some type of the process, which are examples. Then that examples are Indira Gandhi National, also Tribal University, also Amarkat, in also Madhya Pradesh. But how to prepare that the total plans, the date, the plant name, local name, family, locality, attitude, latitude, longitude, habitat, collector, name, notes, permanent herb description of plants like a root, stem, leaf, flower, color, etc. Also got a specimen name or specimen number. These are a complete process for study of the preparation of the herbarium. Then last ninth one is also filling of specimens, mounted, labeled, and tried to kill insect. Pest specimens are finally incorporated in herbarium where they are properly stored after valuation. After valuing examination, that part of the herbarium which can properly stored, which we can use of, say, insected kill resistant or pest resistant, which parts are which can spray. The herbaria arrange specimens alphabetically according to family, genus, and species, and follow the particular system of classification, which system only Bentham and Hooker classification basis to preparation of the herbarium only means that is called a fundamental process for the or practically which can easily identified system, that is called a Bentham and Hooker classification. So then taxonomic literature, we are a collected taxonomic